Mesdames et messieurs, chers et chers collègues, chers et chers amis, c'est pour moi un plaisir de vous accueillir à la Faculté de médecine pour cette leçon inaugurale du professeur Timothy FRAYLING, un spécialiste de la génétique humaine qui a commencé sa carrière au sein de la National Health Service du Royaume-Uni, où il suit une formation de généticien moléculaire, diagnostique dans le cadre de la prédiction des risques dans les familles atteintes de mucoviscidose et de dystrophie musculaire. Il rejoint ensuite l'université d'Exeter et obtient en 1998 un PhD centré sur l'identification des mutations dans les gènes causant des troubles monogéniques du diabète. Il va ensuite gravir tous les échelons académiques et est nommé professeur ordinaire en génétique humaine en 2007. Il est également chercheur invité au Centre national de génotypage d'Evry, ainsi qu'à la Faculté de médecine de l'Université de Genève. Ses recherches sont axées sur des approches à l'échelle du génome, visant à identifier les variations génétiques associées à des maladies humaines courantes ou rares, et à traduire ces connaissances en une meilleure compréhension de la biologie des maladies. Il a ainsi identifié des liens entre le développement de l'enfant et le diabète de type 2, ainsi que des variations génétiques liées à des risques accrus d'obésité et des risques moindres de maladies métaboliques. Il est nommé professeur ordinaire du Département de médecine génétique et développement de la Faculté de médecine de notre université en novembre 2023 et est titulaire de la chaire Fondation Louis-Jeantet de médecine. Il dirige une équipe de recherche pour étudier la génétique et la génomique des maladies métaboliques courantes en se concentrant sur le diabète de type 2, l'obésité et les affections connexes. Pour cela, il combine la génétique humaine, de grands ensembles de données, avec notamment le séquençage du génome de 100 000 personnes, et d'études cliniques pour comprendre les causes et les conséquences de l'excès de poids malsain. Fort de son riche parcours, le professeur FRAYLING travaille depuis son arrivée à Genève à la mise en place d'une biobanque d'ADN en collaboration avec le département diagnostique des hôpitaux universitaires de Genève, ainsi qu'à la construction d'un projet national intitulé Genome of Switzerland, en miroir au projet européen Genome of Europe, visant à créer une base de données suisse de référence en génomique, et ce, en collaboration avec l'initiative Health 2030, portée par les institutions académiques et hospitalières lémaniques. On le voit, le professeur FRAYLING est déjà bien intégré dans la dynamique locale, régionale et nationale. Je vais lui passer la parole en vous rappelant qu'il fera sa présentation en anglais, et ce, pour ne pas diminuer la qualité de son humour très british, qui transparaît déjà d'ailleurs dans le titre de sa présentation, qui en a intrigué plus d'un. Et donc, Tim, it's really an honor and a pleasure to have you with us at the University of Geneva and look forward to your talk. Thank you very much.
Merci beaucoup, Antoine. Donc, je peux peut-être essayer de parler quelques mots en français parce que je suis en train d'améliorer mon français. Mais quand j'ai dit peut-être je peux faire la présentation en français, les administrateurs m'ont dit non, Tim, il faut parler anglais. Il faut parler anglais. Donc, so I will speak in English, and then maybe if I do another inaugural lecture next year, I can do it in French when my French improves. So, let's just switch to the title. A little bit of fun to start with. I wanted you to explain a little bit about the title to my talk. This is a famous poem from a famous British poet called Philip Larkin, who I think wrote this poem in the 1970s. And they said, the poem is, "They mess you up, your mum and dad. They may not mean to, but they do. They fill you with the faults they had and add some extra just for you." I don't think he was thinking about molecular biology at the time, but I thought it was rather nice because clearly it reflects what subconsciously we probably all know, and that is that we inherit some things from our parents that they didn't have much control about, which is the things in their genes, but they also have some control over how we grow up. Incidentally, this isn't the actual word he used. If you Google the poem, he used a word that I didn't want to repeat in public. So, I have three broad topics to take you through over the next 35 minutes. And the first is that we are truly in a really exciting era for the use of human DNA and genetics in studying human disease and applying it to medicine. And I'll try and emphasize why I think that's the case over the next few minutes. And then I'll turn to some examples of how we've used human genetics over the last few years to understand two diseases that I have been studying most of my career, which is obesity and non-insulin dependent diabetes, or type 2 diabetes. And then at the end, I'll just touch on, a little bit on the topic of personalized medicine, which is slightly different to some other aspects. So personalized medicine is very much about taking your DNA sequence and doctors being able to say, right, I'm going to treat you differently based on your sequence, which is just one aspect of human genetics. So I was very lucky to start my genetics career in 1993, and my career has spanned really a large chunk of an era when we've been able to link genetic information, DNA sequence information, to human disease. So the cystic fibrosis gene was cloned in 1989, and that was really the first example of a single gene disorder that we could really analyze in the healthcare service. And I started my career in Oxford looking at 4 mutations, so just four positions in the genome out of the 3 billion positions. We were able to analyze and test people to see whether they had inherited the cystic fibrosis mutation. And really looking back, it's an extraordinary advance in what we've been able to do since, because we can now look at all 3 billion letters of the human genome in hundreds of thousands of people. Just to go back to basics to start with, the study of human genetics is conceptually very straightforward, very simple. We have this code consisting of 4 letters. Get this up a minute. So we have this code consisting of 4 letters, A, C, G, and T, and you can think of it as a simple long line of information. It just so happens it goes up to 3 billion, and about 1% of that, so only about 1% of that code, actually encodes the proteins. Those are the genes. Those are the sequences which actually encode how you build a protein, which goes into building a body. And 99% of it is non-coding, but has important information about how genes are switched on and off. And that sequence, that basic sequence for a human being, was almost completed, or pretty much completed, about 20 years ago. We've been sort of filling in gaps and tidying it up more recently, but the basic sequence was known about 20 years ago. But what we're really interested in is the variation. So in one position where someone in one copy of the genome may have a C and another copy of the genome may have a T. And what we want to do is very simply in disease, and this applies to most of what we do, we want to take people who have 2 copies of the T and people who have 2 copies of the C and people who have 1 copy of each, and we want to measure their risk of disease, or we want to measure levels of something such as cholesterol levels or your body mass index. And we simply use statistical tests to say, right, do these people look different from these people? And are the people with 1 copy of each somewhere in the middle? If we were looking at recessive disease, we might just be interested in whether these people had a higher risk than both of these groups. But if you've got that concept, you're 90% of the way towards being a geneticist. That's it. However, the technology has been such that over the last 30 years, it's not been possible to read all of that, as I mentioned. So really, before 2007, we were limited to reading certain positions in the genome, maybe a few hundred positions, and sequencing 1 or 2 genes at a time. It was a bit like having a pair of binoculars to look at the universe. We could find a little bit of information, but far from all. In 2007, the technology changed quite dramatically in that with microarray technology, we were able to look at common genetic variants spread across the whole genome. So about 1 million positions in the genome that are commonly varying. And we were also, as time went on, able to increasingly sequence that 1% of the genome that encodes the genes. So we now have a more powerful telescope. But it's only been really incredibly recently that we've been able to read that full 3 billion, the 6 billion if you count the 2 copies, of the code in really large numbers of people at what you might call population scale. And as literally as recently as November 23, so as I arrived in Geneva, data has been released from 750,000 people from the UK and from the USA, with all 6 billion letters read, and with extensive information about their disease phenotypes. So it's really an incredibly recent increase in information about the human genome. So we now have the powerful observatory to look into the universe. This genetic information and molecular information and its advance in our ability to measure and understand it also comes alongside an increasing ability to look at people's disease. And this really comes from the fact that most hospital and healthcare systems have been recording what their patients have and the diseases they have in electronic medical records since the late 1990s or the early 2000s, and that's true for the Hugen here as well. Which basically means that if you can take a blood sample from your patients coming through your healthcare system and link it, and you have the informatics systems which are able to link it back to samples and genetic information, you effectively have a human study, a human laboratory, if you like, of people with their data from the 1990s or early 2000s. And potentially, if you're young enough, you can follow these people up until they die in many decades since. So this is a very powerful, and it's not perfect. There are issues with defining disease in medical records that mean it's far from perfect, but it's a relatively cheap way of obtaining a huge amount of information about people's disease and creating what you might call a long-term human laboratory of disease information. So as I say, this we can think of this as a bit like a human lab, and 2 ways of doing human genetics, or 2 ways of using human genetic information. The traditional way is forward genetics, so for many of you working in laboratory systems, you might be taking an in vivo model such as a mouse or a zebrafish or a Drosophila or cells, and you might be spending your time doing extensive phenotyping, studying a particular system. And then if you find something interesting, you might be interested in finding out what the human genotype and human phenotype is. Reverse genetics, in contrast, is where we can start with the humans. And this is what we can do now with much more power. We can start with people who have missing a gene or have a particular alteration in a gene. In many cases, we'll have extensive pre-existing phenotype information from those individuals, from, as I say, medical records, but also large biobanks and research studies. And then if we find something interesting in people, and we can't do all of the experiments we want to on people, obviously, we can then take it into more detailed model systems. So this really sets us up as starting with human beings to study disease. Importantly, this is not just about studying genes themselves. We can also study in great detail the other 99% of the genome, much of which is important in controlling how genes are switched on and off, which cells and when and where in development. And during the course of our lives, those genes are switched on, and we call many of those elements things like enhancers, because they enhance when a gene is switched on and off. And because we now have so much genetic information, we have a variant pretty much every 4 base pairs, 4 letters across the genome, so we can really have a genetic readout of what's going on in your regulatory regions as well as what's going on in the gene themselves. An example published by, unfortunately not myself, but I can take about 0.1% of the credit, because this paper was published last week by one of my former PhD students, John Perry, who actually left Exeter many years ago, so I really can't take any credit at all. But he's now a professor in Cambridge, and and he released this very nice paper that illustrates my point, hopefully illustrates my point quite nicely, and that he used the genetic information from the 740,000 people, all 6 billion letters, and he found that approximately 1 in every 4,000 people have a mutation in the gene that means they're effectively missing one of these 8 genes, or at least missing one copy of these 8 genes. And you can see there's only 100 or 200 people with each of these variants. And what you can see on the x-axis here is the difference in body mass index for these people who are missing these genes. So very roughly, you can see there's a difference of about 4 body mass index units. So that's like going from a BMI of 27 to 31, or going from 65 kilograms weight to 77 kilograms weight for someone who's average height. So substantial effects. Importantly, only 2 of these genes were really known before from animal model studies and from what we knew about the biology of obesity. And 6 are brand new, discovered this year. Incidentally, rare events happen in lots of people because there are populations consist of millions. Things which only occur in a few thousand, or 1 in a few thousand people, 1 in 5,000 people, 1 in 4,000 people, still means there's lots of people walking around with these genetic changes which are affecting their health. And just roughly looking at the Swiss population, this would be 800, with these particular genes I've just described, this would be 800 people in the Canton de Genève and 13,000 in Switzerland who are walking around with this genetic change, which means their BMI on average is about 4 units larger than average. Paper came out last week, so we don't really know how we would treat those people differently, of course, but that would be a starting point for studying those people and their condition. So turning to a couple of examples from my work, and a little bit more about the genetics of obesity and body mass index. This is something that some people struggle with, even clinicians and scientists, because after all, we have the same genes as our grandparents and our great-grandparents pretty much, and yet overall the population was a lot slimmer in those days. And what you can think is that the shift in the average BMI and the distribution of BMI in the population over the last 50, 60 years has been entirely environmental, as our lifestyles have changed. But actually where you are on the scale of BMI is substantially genetic, probably somewhere in between 30 and 40% of where you sit on the BMI scale in the population is down to what you inherited from your parents. And of course, you're probably familiar with the classic way of trying to quantify that is from twins. So we compare BMIs from people who are identical twins to those who are non-identical twins, and sure enough, you see that the BMI of the identical twins pairs is much closer than those who are non-identical. Twin studies are far from perfect, small numbers, and maybe parents are treating their identical twins differently to their non-identical twins. And we can actually, now we've got genetic information from the whole genome in lots of people, we can do more sophisticated experiments. So for example, I could take everyone in the room, I could take a blood sample, I could measure your genetics, and I could find out how related you all are to each other. I could find out if some of you are cousins with each other. Probably some of you in the room are 6th, 7th cousins, right? And I could tell you that information, and I could quantify it, and I could say, actually, you're 1%, you share your 1% of your genes with the person sat next to you, you share 2% of your genetics with another person in the room. And if I use that information, sure enough, that those of you who share 2% of your genetics will be more similar in BMI than the people who share 1% of their genetics. So that's another way that we quantify the heritability or the genetic component to a disease. So I was very fortunate to be involved in a large team of researchers in the UK who in 2007, when this new technology came along to enable us to scan the whole genome for the common variation, we found this genetic variant on chromosome 16. This is a plot of the genome from chromosome 1 to chromosome 22, and on the y-axis Each dot represents how strongly a genetic variant is associated with BMI, with type 2 diabetes in this case. And we found that these variants associated with type 2 diabetes, all these ones here, were altering things like beta cell function, how much insulin you produce. This one here was actually associated with BMI, and because people with diabetes were heavier than people without diabetes, this signal came up. And when we looked at it in more detail in children aged 9 years who'd been scanned, we found that this gene truly was the fat gene, and that children were with particular copies of the gene were more than 10% heavier in terms of fat mass compared to those who didn't have a particular version of this variant. And this is me with my mentor at the time, Andrew Hattersley, carrying the amount of fat that these people have compared to these people, for a nice shot for the media. Since 2007, what's happened across all common phenotypes and common diseases pretty much is that we've been able to identify typically between 100 and 1,000 common genetic variants which are robustly associated with your disease, your favorite disease or your favorite trait, and BMI and type 2 diabetes are no exception. And this allows us to quantify or to give everyone what we call a polygenic score. So we can add up all the genetic variants you have that are associated with BMI or diabetes, and we can put you on a genetic risk. So risk scale. So put simply, if we had 100 genetic variants associated with BMI or a disease, we can give everyone a score of between 0 and 200 because you have 2 copies of everything. Some people could have 200, some people could have 0 because it follows a Gaussian normal distribution. Most people are in the middle, but you can think of these people at high risk and these people at low risk. When we do this for BMI and we look in children, we see that the genetics of BMI overall seems to establish itself very early. So this is a series of plots from the same children or largely overlapping children from a single study in the UK, a famous study called the Ausbach study. Here are children at birth. Here they are 18 and ages in between. And what you can see on the each x-axis is deciles. So here's the 10% of children and their weight the 10% of children who have the highest genetic risk of BMI. Here's the 10% who have the lowest genetic risk of high BMI. And you can see at birth that there's not really much effect there, and that is because most of your size at birth is governed and influenced by your mother. And then you can see as you go through the years, there starts to be an influence of your own genetics, and by 3.5 years, perhaps when children can open the fridge and have a bit more autonomy over their calorie intake and outtake, you start to see an effect such that by 18, just based on your genetics, if you're in the top 10%, you'll be well over 10 kilograms heavier than, for an average height, you'll be well over 10 kilograms heavier than if you're in the 10% lowest risk. So this turns me to one of the neat experiments, one of my favorite experiments we've been able to do with human beings recently, and this is about uncoupling excess weight from its adverse metabolic effects. So on average, people who are more overweight are metabolically less healthy. Their insulin levels, their glucose levels, their fat levels, lipid levels in their blood, their ectopic fat, so the fat in your liver and around your pancreas and heart will be higher. But it varies. Some people of the same BMI can be relatively protected from that, and some people very severely affected. And so what this slide shows is a plot of, or if you look at the left to start with, this is a plot of a genetic variant, an allele or version of a gene's effect on BMI and its effect on type 2 diabetes. And you can see a pretty strong, decent dose-response effect in that the genetic variants that increase BMI tend to increase the risk of type 2 diabetes. Nothing new there. But what you can see here is there's a handful of genetic variants that appear to be increasing BMI, but lowering the risk of type 2 diabetes. And in fact, when we look more closely at these genes, we see that one of these genes is well known. It's a genetic variant in a gene called PPR gamma, and PPR gamma, The protein is an important transcription factor. It switches on pre-adipocyte cells, so cells which are about to make fat cells, and it helps you make more fat cells. And what you can see is that the variant that increases your body fat percentage reduces your risk of type 2 diabetes. And indeed, when we started looking in more detail at more of the genetic variants associated with BMI, we started to find these heterogeneous effects of alleles which increase BMI on type 2 diabetes and measures of metabolism. So here's a classic set of genetic variants here, and the red here shows higher values and the blue shows lower, but this is 38 variants, each row a variant. Here are the alleles which increase body fat percentage and BMI, and these, as you would expect from what we know about excess weight, they reduce your HDL, your good cholesterol, they increase your triglycerides, so the fat circulating in your blood, and then these are markers of fat, particularly ALT and AST are markers of how much fat you have in your liver, and so these genetic variants go with excess fat in the liver. This work was done by two of my team, Susie Martin and Hanya Yakutkar, and what they found, really interestingly, was that we have a similar number, or we could find a similar number of variants which have the opposite effect. So this favorable, what you might call metabolically favorable effect with genetic variants that increase alleles which increase body fat percentage and BMI, but they're increasing your good cholesterol, they're lowering your circulating fat, they're lowering the amount of fat you have in your liver, which is interesting for lots of reasons. The biology of each one of those genes is going to be really interesting to look into. We thought it would also be interesting to look at what the consequences of carrying those variants are on lots of diseases. So if you have high BMI, you are not only at risk of type 2 diabetes, excess risk of type 2 diabetes and heart disease, you have a higher risk of osteoarthritis, you have a higher risk of thrombotic events, asthma as well, heart failure, and indeed many cancers. It's not healthy to be overweight. But what we thought was we can do a human experiment. We can uncouple the metabolic, using these genetic variants, we can uncouple the metabolic effects from the excess weight. And we can say, well, is it the excess weight itself, which you can imagine might be more of a factor in something like osteoarthritis, where the weight on your hips and knees could be more important than the adverse metabolic consequences. Or is it more metabolic? Is it that it's not the excess weight itself, it's the adverse metabolic consequences? And so we might expect this sort of pattern with the genetics of metabolically unfavorable adiposity increasing risk and the genetics of favorable adiposity decreasing risk for conditions such as diabetes, but for conditions such as osteoarthritis, the genetics of the genetically favorable weight, excess weight, is also increasing your risk. So we did this and looked at lots of diseases. Here are the what we kind of knew were metabolic diseases, so positive controls. And you can see, as we'd expect, metabolically unfavorable adiposity increasing risk, favorable adiposity decreasing risk, not just of diabetes, but polycystic ovary syndrome, stroke, gout, etc. It gets more interesting when we look at some other conditions. So here we have patterns with heart failure and atrial fibrillation, where the favorable adiposity genetics is more ambiguous. And one hypothesis here is that the excess weight itself is an excess need for vascularization, is placing more strain on the heart. And there's a really fascinating relationship between breast cancer and excess weight. And other studies have shown this, that excess weight from an early age, so in girls and young women, that can actually have a protective effect on breast cancer. And that's what we see more with the metabolic effects here for reasons we don't know. It's a topic in itself. Also gets more interesting when we look at some of these conditions where the results are more consistent with some sort of mechanical or load-bearing effect, as you see the arthritic conditions, excess weight, metabolically unhealthy weight, metabolically healthy weight still increases the risk of osteoarthritis and rheumatoid arthritis. We think it's pretty interesting that the thrombotic conditions seem to be also more about the mechanical, presumably the mechanical effects of excess weight rather than the metabolic consequence, or as well as the metabolic consequences. We plan to follow this up, as Antoine mentioned, by looking in more detail at fat cell morphology in human beings. There is some precedent for what might be going on here from animal models. This is a fat mouse, by the way, and this is a very fat mouse made with by overexpressing this gene called adiponectin. But this mouse is actually healthier than this mouse. So this is an example where we did know something about what might be going on from animal studies. And this mouse has more but smaller subcutaneous adipocytes, subcutaneous fat cells, which we think is consistent with it being able to store fat subcutaneously under the skin rather than around its organs. So we don't plan to do exactly this with human beings, but we will hopefully recruit individuals with the collaboration of the Centre de Recherche Clinique and to take fat biopsies and study their fat cells and link it to their genetics in more detail. We also incidentally have done a similar experiment separating or uncoupling childhood BMI from adult BMI and see some very interesting differences between excess weight when you're a child compared to when you're an adult. So just a second experiment, human genetic experiment, if you like, and this is a more general one, in that since 2015, the pharmaceutical industry have really realized that human genetic information, linking genetic information to phenotype information and disease information, is a really powerful preclinical tool to use to help prioritize drug targets, to help validate them, and to help repurpose them. And there's a very nice example that comes from something that's very topical in the news at the moment, which is the use of these drugs, the most common one being semaglutide, which have been used to treat diabetes for some time, but now increasingly are being used and sold directly to the public because they help you reduce your weight. But basically, the principle behind this is another very simple concept of human genetics, in that if you have a genetic variant in gene B and a genetic variant in or near gene F, the protein, and if those genetic variants are associated with type 2 diabetes, the protein products of those genes are more likely to work as a drug for type 2 diabetes. Doesn't mean to say they will work, because most drugs, when they enter the phase 1 pipeline and the development pipeline, they still fail. Only about less than 10% of drugs that start clinical phases actually succeed. But the genetics can help move the dial. It can help tell you how likely it is to work. And if there's no genetic support these days, it's less likely to work as a drug. If there is genetics, it's more likely to work. And so when it comes to, and there's very nice proof of principle from the weight loss drugs and the diabetes drugs. So these are drugs, semaglutide and tirzepatide, which mimic these hormones which are produced by the gut, the so-called incretin hormones that were produced naturally and basically produced from the gut. They signal to all sorts of places, not just the pancreas mainly, the pancreas to secrete insulin, but all sorts of other places, the stomach, the brain, basically to tell the body if food is coming, slow down, be ready to secrete insulin, be ready to slow down your food intake and monitor your stomach emptying, etc. And these drugs, and there's these two receptors, two hormones and their receptors in particular, GIP and GLP, that are produced from slightly different cells in the gut. And these drugs mimic these effects. And indeed, it looks like the one which targets both, including the GIP receptor, has a bigger effect on weight loss. And sure enough, we have genetic variants in these genes. And so we can, and actually we found that one of these variants, particularly the one in the GIPR receptor, we found in 2010, so long before the drug industry or the public had been thinking of using this outside of diabetes. We showed outside of diabetes that a genetic variant in the GIPR receptor is altering weight. Interestingly, with GLPR, less clear effects, which maybe points to the drugs being working better through the GIPR axis. This is a slide which shows another nice example of that. So the GLPR variant actually has a bigger effect on diabetes, and this was a study, not my work, but this was a study that did a proof of principle what you might call a pharmacovigilance study, where they showed that a genetic variant which lowered your type 2 diabetes risk was not associated with excess heart disease. It was very important. It's still very important for all diabetes drugs to be checked for cardiovascular safety. The predominant reason we treat type 2 diabetes is to reduce the risk of heart attacks and cardiovascular events. So if you give someone a drug and it helps their diabetes but increases their risk of heart disease, it's no good at all. And this genetic, and sure enough, the trials of the drug showed that they also reduced heart disease, but the genetics also mimicked that role. And then also showed no sign of adverse events with cancers or neurodegenerative diseases. So you can use, up to a point, there are differences, but up to a point you can use genetics as cheaper mimics of what might happen in a trial. So I will now just turn to the final example, which is that of personalized medicine. So in contrast to what I've just described, which is really about using genetic information in everybody and trying to make insights about biology, personalized medicine is very much about taking your DNA sample and saying, right, your doctor needs to do something different with you because you've got this particular change in the DNA, or this particular combination of changes in your DNA. And so up till now, really, this concept of personalized medicine has really only been used in healthcare services for rare disease. So if you have a mutation in cystic fibrosis gene or muscular dystrophy, lots and lots of rare diseases. Very nice example from my former place of work in the UK, where my mentor discovered neonatal diabetes. So diabetes when you're born does not need to be treated with insulin. They can be treated with an oral agent. So lots of examples from rare disease, but in common disease, it's not really being used extensively for common disease. An exception is cancer, where we sequence tumors and we can make decisions about which drugs are going to work in tumor profiles. But in terms of sequencing the genetics that you inherit from your DNA, not used so much yet in common disease. I would argue that we need to be seriously thinking about doing that, and indeed many countries and healthcare services are thinking about that and introducing it. And there's probably many applications actually, but just picking out three. Improving risk prediction. So doctors treat and diagnose disease, but they also spend quite a bit of time and money trying to prevent disease. So cardiovascular events, often we're being screened for things, and if we're at high risk, we might be put on statins, even if we haven't yet had a heart attack. Lots of screening programs in cancer. Very topically, children who are at very high risk of type 1 diabetes, we may be able to delay their type 1 diabetes with a new drug that's been approved for that purpose. But you need to know who's at risk. Pharmacogenomics, so about 90% of you in the room will carry a change in your DNA. That will mean you will not respond to a commonly used drug, or that you will have a higher risk of having an adverse reaction to a drug. And healthcare services are introducing some of those genetic tests, including here at the Hug, for those purposes. And then there's something I've learned about fairly recently, and that's what you might call personalized biomarker ranges. So doctors spend a lot of time measuring stuff from your blood. In particular, it might be your blood cell counts, your hemoglobin, your cholesterol levels, markers of kidney function. Most of the time, not all the time, but most of the time, those things are just consequences of disease. They are not causing the disease. So a good example would be cystatin C and creatinine. These are critical measures that are used to measure how well your kidneys are functioning. But if you have altered cystatin C levels or creatinine levels, it has nothing whatsoever to do with whether you will get kidney disease. It doesn't cause kidney disease. And yet these things all have a genetic component, sometimes quite a substantial genetic component. So the level at which you're measured might not be right for you. And there's actually relatively straightforward ways of correcting people's levels based on their genetic background. I will just say a few words about the first example, though, the risk stratification, because I think this is where, well, alongside pharmacogenomics, I guess, but this is where things are most mature. So here's a classic patient. This is me, male, non-smoker, aged 51. Father had a heart attack, 67. BMI reasonably slim, slightly high cholesterol. This is too much Swiss cheese and French cheese and chocolate. I think it's very hard to reduce that. But you know, there is American Heart Association criteria saying that people in my age range should be screened. We should be measured for our 10-year risk of atherosclerotic cardiovascular disease. And this slide shows why we should do that. This is the people binned into groups based on traditional risk factors. So here, this group here will be enriched for younger, female, non-smokers, people with low blood pressure, people with low cholesterol levels, and this is their 10-year risk of a heart attack is very low. And then here are your older male smokers, high blood pressure, high cholesterol levels, and their risk of having a heart attack or a stroke in the next 10 years is much higher. What's on the x-axis of each of these graphs, though, is the genetics, and how the genetics pulls the genetics of heart disease pulls people apart. So if you just take people who are already at high risk from all the traditional risk factors, you can see that if you look at the blue line here, this is people of European ancestry. The people who are at lowest genetic risk have a much lower risk, about 5%, compared to people who are at the highest genetic risk, who have about 15%. As a rough rule of thumb, for most common diseases, the genetics now will explain about the same amount of risk for that disease as LDL cholesterol does for your risk of heart attack. So the genetics is doing the same as LDL cholesterol for heart attack. And importantly, you only need to measure it once to get all the information from all of the diseases. And it's complements those, so it's not overlapping with, or overlapping very little with a single measure of LDL cholesterol level or your blood pressure. Everything I've spoken about so far, or the vast majority of it, has been about research from my research and others using large-scale human genetic data sets from the UK, from America, from Northern Europe especially. And so I just wanted to finish on a positive note by saying something about opportunities in Switzerland. And that, I think, having moved, I was very excited in November because the opportunities are strong. I think Switzerland is well set up to have its own human laboratory, if you like, in that 10% of the Swiss population have signed a general consent for their samples and medical records to be used for research. As a percentage of population, I think probably only Estonia can compete, and maybe Denmark can compete with that. And of those people, you know, Geneva is very well set up. More than 110,000 people in the canton have signed the general consent. And then with Antoine's support, as he said earlier, we are in the process of setting up the hospital informatics. That means we'll be able to intercept blood samples from people who've signed the consent and store the blood, spin it for plasma and serum, make DNA, and be able to do that genetic component to disease. And then critically, we have and this is where Geneva is ahead, I think, have a maturing medical record linkage process, which means we can link the genetic information to the disease information. And hopefully, over the next, I've had a fantastic 30 years career, and hopefully I've got another fantastic 15 or so years helping this happen. And then to finish on the most important slide, really to talk about my work from now, and that really, really chuffed and pleased that I've appointed these fantastic early career scientists. Aurélie, Lorik and Lisa have made a fantastic start. We are open to collaboration. We have 740,000 whole genomes in our computers on the cloud. We are analyzing them as we speak. And so we're very much open for collaboration. We will be able to find 100 people with a rare disease of your interest, or we will be able to look at common disease. If you're a clinician interested in the clinical aspects, if you're a laboratory scientist studying a very particular piece of biology and you want to know what the human phenotype is, we are here to help to do these bits in green, and very much open for collaboration. So I will stop there by thanking. We already have some funding from some local Swiss charities and from Novo Nordisk. I forgot to say at the start, but most importantly, the Fondation Louis-Jeantet, who are sponsoring me and the team. Fantastic, and thank you very much. And I'll leave you to contemplate whether you chose your parents well or not, or whether your kids have chosen their parents well or not. Thank you.
Well, thank you so much for this exciting talk, for the spirit of it, the openness to collaborations, and showing a new way to think about human genetics and research. So that's really exciting. I think we have 5 minutes if there are burning questions in the audience, knowing that, yes. 1, 2, 1, 2. Yes, it works.
Thank you for a beautiful talk. I was wondering if I should choose my parents differently in times of starvation or famine. In other words, are those obesity alleles also protective from dying of starvation? Is there any evidence for this in populations?
Yeah, good question. In a short answer is not really. We don't know enough about the genes to say are they there because they've been, or the versions of the genes, the alleles, are they there because they've been pushed up to higher frequencies to help us eat more? We know that most of the genes discovered so far, where we know the biology, the biology acts in the hypothalamus, so the part of the brain that really controls appetite sensing, so controlling when you're full. And we know those genes are that effect is seen in childhood, but that's partly because that's only where we've been looking. And some of those new genes that I spoke about, they don't appear to be affecting children in this. I glossed over that because I didn't have time, but they don't appear to be affecting BMI in kids. They appear to be having later onset effects. There are ways of looking at the genome and saying, has this variant been swept to higher frequencies by selection? Generally speaking, those sorts of things are only detectable for variants which have really strong effects on infectious disease. So the CCR5 variant swept to much higher frequency in Africa because it protects you from HIV infection, for example. With appetite and eating, it's harder to see those sorts of things in the genetics.
Just from walking around, it looks like there are more than 800 obese people in Geneva. So what are the other ones?
So this is all about
victim.
So the 800 example would be people with a knockout or one copy knockout of one of those new genes. Most of us vary in body mass index, and most people at the end of the scale will have a complex mixture of high genetic risk and high environmental risk. So the genetics is 30 to 40%, which means there's still 60 to 70%. We know that poverty and where you are on the socioeconomic scale has a big effect as well. I'm not claiming genetics is only part of it.
That worked. Thanks so much. I think my question is quite related to previous question, but maybe let me phrase it differently. So, because you presented this as we are now looking at this 5% or of the normal distribution, if we go to the lower 5% of the distribution, Because, you know, to be a bit provocative, we could also consider those to be the odd ones out, right? In our current society or culture, we have evolved in environments where we optimize and maximize our energy intake and to lower and minimize our energy expenditure. And so that's why we end up with the culture that we have. And so you could also argue that the default is that we increase as much as our body weight, right? As much as we can. Then what makes very slim people stay slim? If that's basically the default setting of our
so
genome.
so the same answer really. You know, there's a complex mix of genes and environment, and some of us are better off than others. If your parents are rich, wealthy, and have paid lots of attention to your health, brought you up well, you know, impressed upon you the need not to do too many risk-taking behaviors, have restraint, and you've inherited good genetics in terms of the risk, then you'll be slim.
But are these genetics always good? I mean, that lower 5% might also have some risk associated to.
So for BMI, and the same goes for cholesterol levels and also for alcohol drinking, unfortunately, there's no kind of, there are, if you look at data, epidemiological data, you do see kind of U-shaped curves with people who are very slim or people who don't drink at all, don't drink any alcohol at all. They're often quite sick, but that's down to reverse causality. So if you're very sick, especially if you're older and you're going into cognitive decline, for example, you actually lose weight. So you see this reverse causality. So we have to be very careful when we look at epidemiological data. That's where the genetics can help, because we are much less biased by that reverse causality issue.
I think it's always a bit dangerous to talk about BMI and calorie intake just before an apéro.
Yeah.
So maybe we should stop the discussion now, and if you really want to continue in this discussion, you can do it over the apéro. Here is the starter kit for new professors in the faculty. I know you've already been carrying the flag of the University of Geneva well, but here's another excuse to do so with the Faculty of Medicine sweater.
Oh, marvelous.
It hasn't been personalized based on your genetic genome sequence, but hopefully it will fit.
Excellent. Thank
It
you
seems
very
really
much.
good. Okay, well,
Thank you.
so much for the talk. Thank you everyone for participating and hopefully enjoying the talk. And now time to enjoy the apéro. Thank you very much. Bye-bye.